Thanks, Bob. Somebody recently said the current situation is like a football game that's gone really bad and you're losing and you walk out in the fourth quarter. And he said the only problem is we're in the first quarter. Anyway, Bob really clearly articulated the challenges we face in science today and as scientific institutions. And I think the question is, what do we do about it? And clearly lobbying both Congress and the public has done some good. But the question is, we don't have much control over that. Can we do something about the current situation that we do have control over, and what is it? And so I'd like to tell you about the experiment we're running at Scripps in that regard. Just probably most of you think you're at the Oceanographic Institute. Okay, we're actually a basic biomedical research institute, one of the largest two independent institutes in the U.S. We're small, we have 120 faculty, but I think we punch above our weight, and we have a top 10 graduate student program. But historically, Scripps has been focused on really fundamental research. And a few years ago, I was asked to take on the leadership of Scripps, and I accurately deduced that that would not be a fun job. And the question is, why would you do it? And I think the answer is if you can do something new and impactful. And so the question that we asked was whether you can actually begin to think about creating a new sustainable model for biomedical research. And you could argue at the time we were thinking about this is what we wanted to focus on. It was actually prescient for today's situation. But we pursued that, and I'll give you a progress report on that experiment. Scripps is a good place to run experiments because unlike most universities, we have a very a board that's willing to take risks to do new things. As we think about new things, you always have to keep in mind that the core value of research institutes is the quality of its basic research. And we want to make sure that continues, and that, as Bob pointed out, is really threatened by the changes in federal funding. But moreover, it's been threatened for the past 30 years in terms of the focus on high-risk funding, high-risk reward research, transformative research. It's harder to get that funded nowadays, so we do need new mechanisms to make sure we do cutting-edge science. At the same time, what we'd like to do is accelerate the impact of that science on new medicines and human health. And we want to do that because we want to accelerate the development of new medicines for unmet needs. But moreover, the taxpayers are the people investing in the science, so we want to give back to the taxpayers, and I think new medicines are something they understand is valuable and the impact of science. And finally, in doing that, can we actually create what I'll call a new business model for nonprofit research institutes that's sustainable? Because as you make new medicines, the value of the medicines goes up as you get into the clinic and you show they work in people. And so if you partner those, all of a sudden you have a mechanism for creating new revenues that you can reinvest in the research enterprise. So every time success begets success, and as you're more successful, you have more resources to invest in the scientific enterprise. So the question is, can you actually create a flywheel-like model to fund basic research? And so we are focused on this idea. Can we do great science, but can we accelerate its transformation into new medicines? And although it's the golden age of biology, we know more about human health and disease than ever before. the development of new medicines has really lagged behind our knowledge of human biology. And the question is, why is that? And I would argue it's because discoveries are made in nonprofit academic institutions. They don't have the experience, the infrastructure, and the processes to turn those discoveries into medicines. So that's done in pharma and biotech, which have very different cultures, very different processes, and very different motivations. And those differences create a real barrier, an intrinsic barrier that slows and even stops the progression from a research discovery to enabling a new medicine. And how do you overcome that? It's called the Valley of Death, and the idea we had was, could you seamlessly go from a discovery in the laboratory all the way to the development of a new clinical candidate, all the way to testing that in human clinical trials and show it works, all under one roof. It's soup to nuts, it's very fast, it's seamless, and it's very efficient. And moreover, when you do get to human proof of concept, the value creation is huge. And the returns are going to be 10 to 20 times the returns that academic institutions normally get. If you have a 10% royalty on a $5 billion drug, you're getting $500 million a year of unrestricted money to fund science and technology. So that was the idea. And the question is, can it work, and how would you do that? So we actually created a translational arm at Scripps. It's called Caliber Skaggs. And it was pretty simple. I had created a nonprofit translational institute called Caliber, which I was the president of. And when I became president of Scripps, it was pretty easy just to put the two together, okay? Nobody really argued with it. So we did that, and it's now a division of Scripps. It's highly collaborative with institutions, with pharma, with foundations. It's only 140 people, so it's small compared to Scripps's research arm, but it has advanced 14 programs into human clinical trials. It advanced 4 programs this year alone in the human clinical trials, which is really unprecedented in a nonprofit research institute. Importantly, the fundamental principle is it operates autonomously. It defines its own programs, it executes its own programs, it owns its IP. So it's completely independent of any control by pharma or others. And the revenue generated from that goes back into the research enterprise. And because we're going all the way to the clinic, you get a 6 to 8% royalty, which is large. And hundreds of millions of dollars in milestones. And just in the past 5 years, we've generated over $500 million in revenues that can be reinvested. So let me give you just an update. The reason this works, one of the reasons is we've developed a lot of technology platforms that really facilitate and make efficient drug discovery and lower its cost, all the way from 160 billion member chemical libraries to the world's largest repurposing collection. And we've used that to operate, to advance a lot of programs across almost every disease area. One thing I've never been accused of as being overly focused, okay? So we work on virtually every disease opportunistically and across all modalities. But the important point of this slide is you're developing a pipeline. The way it happens today is somebody has a great idea, they go to venture investors, they raise money, they hire a CEO, a chief scientific officer, they find a site, they raise the money, they hire all the people, and if they're successful, they partner it with pharma, and then you start all over again. It's very inefficient. Why don't you just build a machine that does it repeatedly and sustainably? So that was the point of this slide. Let me give you some examples of the kinds of things we do. If you're going to do this in an academic nonprofit setting, you really don't want to compete directly with pharma because they have far more resources. So you want to do things that are new and different. And so if you think about what drugs do today, most of them just slow disease progression. Very few stop it, but none reverse it. So can we make a whole new generation of drugs that create a new paradigm in medicine where you can actually reverse disease? And so the biggest killer in the world is heart failure. There are 2,200 heart attacks every day in the United States alone. When you get a heart attack, part of your heart dies, okay. We now keep people alive. We extend morbidity and mortality, but by not much. But you progress into heart failure, and sooner or later your heart fails. And we can't stop that. So the question is, can you reverse that? So heart doesn't have stem cells, but there's a pathway involved in the development of the heart you can turn on, the YAP pathway, which we did with a small molecule. And when we give a pig a heart attack, okay, which is shown in the upper right, blue is normal heart, red and yellow are dead heart. You see that after 3 days, if you image the heart, you kill roughly a quarter of the heart. If you wait 30 days, you're in full heart failure. Most of the heart is dysfunctional. If you take that same pig, give it heart failure, wait 3 days, you see the middle panel on the lower slide, you have a lot of cardiomyocyte death. If you give one treatment of the drug to that pig and come back 30 days later, you have normal heart in every way, by imaging, by ejection fraction, by cardiac output. So you get complete reversal of the disease. And you can actually see that on this slide. If on the left side is a pig where you cut off the blood supply to the left ventricle, and you see the bottom left quadrant of the heart doesn't beat, it's dead. If you give one treatment of the drug and look at the heart, completely restored function, okay? And pigs are a great model for a human because you can transplant a pig heart into a human, okay? So we're gonna put this into people in about a year. And so it may actually stop heart failure. There's 8 million people in the United States with heart failure. But the other question is, I don't have heart failure, but if I take it, will I get a 40-year-old heart? That's what I'm really interested in, okay? We've done the same thing with lung disease. Respiratory disease is the third leading cause of death in the world. The lung does have stem cells, and you can actually find molecules that proliferate those stem cells and make more, and then they turn into more lung. So we've made a drug out of that you inhale once a week. It's right now in a clinical trial in Germany at the Fraunhofer Center. And in about 3 weeks, we'll go into IPF patients, fibrosis patients. We have yet to see an animal model of disease that we don't make new lung and reverse the disease. So I think you can do the same thing for lung. We're working on eye with RPEs, and we're working on gut to repair the mucosal barrier. Cancer, one of the most exciting advances in cancer is CAR T therapy. Here's where you take T cells that normally attack infected cells and re-engineer them to target cancer cells. It's almost been curative in blood cancers. It's really remarkable. But it hasn't had an impact on solid tumors because it's a living drug. You're putting T cells, live T cells, back into a patient, and you can't control them. If I have a headache, I take 3 Advil. If it doesn't work, I take 4. I rarely go to 5. But you can't do that with a living cell therapy. So we've invented a way to break that cell therapy into 2 pieces with an adapter molecule that you can then dose. And that works just as well as a normal CAR T cell, but we can control it, so the safety is far better. And now this is in human trials in breast cancer patients. Bladder cancer, it's a disease you get when you get older. The way you treat it is with BCG. It's basically you put mycobacteria into your bladder. It was the first immunotherapy. It doesn't work in about 40% of patients, and it's very painful. But the correlation of its efficacy is correlated with the release of a cytokine called type 1 interferon. And the best way to stimulate that is by activating what's called the STING pathway. So we've made molecules that activate the STING pathway. And we think we can give them intravesically once a month and actually replace all of BCG. And we think in combination with recent advances, this will be curative for that cancer. You know, the biggest advance in cancer therapy has been checkpoint therapy, okay? Actually overcoming the barrier that prevents the immune system from attacking cancer cells. And that's Keytruda. It's $50,000 a year and you need an infusion. So 85 to 90% of the world can't have access to it. Okay? It's the second leading cause of death in the world, and we're talking about health equality. Most of the world can't get checkpoint inhibitors. So in partnership with Gilead, we actually now have an oral checkpoint inhibitor. It's a pill you take once a day. It's cheap. You don't even need a biopsy, you just take it. And if you're the 40% of patients that have checkpoint, where you have PD-L1, it will actually treat the disease. So we think for the first time, we can begin to make the best cancer therapies available to the world. This is one I'm really interested in. Probably nobody here takes Botox. But it's used for cosmetics. It's amazing. It's a safe toxin. It's got two pieces, a heavy chain that targets it to the neuron, and a light chain. There's a very selective protease that cleaves a protein involved in transmitting neurotransmitters. But it's safe. And so we asked, could we re-engineer this? This Botox goes to sensory neurons, which are the pain-sensing neurons. And there's a protein in those neurons that if you have a human mutation in it, you don't feel pain. You can put a knife through your hand and not feel it. And so we're almost done with re-engineering the Botox protease to cleave that channel that's the pain sensor, okay? So now instead of getting a Botox injection to remove wrinkles, if you have back pain, Botox, okay? Joint pain, Botox. Burn, Botox. Surgery, you don't need anesthesia, Botox, okay? And it's non-addictive, and you keep your peripheral pain sensing so you won't burn your hand on the stove. So we're really excited about this as a way to change. We're also working on diseases of the developing world. There's no financial return in that. I'm kind of hopeful that there's a St. Peter and it'll give me a story to tell, okay? But so the biggest killer, infectious disease, is TB. Kills 1 million people a year. 1/4 of the world is infected with TB. The drugs are terrible. It takes 9 months to sterilize the disease. If you're taking drugs for 9 months and they're terrible drugs, what do? You quit taking them, you get resistance. So the big goal of the Gates Foundation, with whom we're working, is to shorten that to 2 months. So we now actually have found a drug that by itself doesn't have much activity, but in combination with a standard of care for TB, looks like it could shorten it to 2 months, which would be really transformative in the treatment of TB. And Gates hopes to get this through approval in 3 years, which would be incredible. How about preventing disease? MMD did a survey of physicians in sub-Saharan Africa. Which would you rather give a patient, a vaccine, a once-a-day pill, or a sub-Q injection that lasts 3 months? And they said the sub-Q injection every time, because with the vaccine, you got to get the boost. You don't know if people are going to take the pills. Just give them an injection at the beginning of malaria season with a drug. It's not a vaccine, it's a drug that stays around for 4 months. They can't get malaria. So we think we've made that with the Gates Foundation, and that's actually right now being tested in Africa. And we've done the same thing with HIV. It's been 50 years since HIV came into the United States. We still don't have a vaccine, and nowadays it's even harder. But Gilead made a drug, lenacapavir, that's long-acting. It's a sub-Q injection with an auto-injector. You get it, and you cannot be infected with HIV for 6 months. 100% reliability. So that's in the clinic now. It's effectively a chemical vaccine for the first time. But it needs a partner, because if you don't have two drugs, viruses get resistant. So we've been lucky enough to make the partner drug for that Gilead drug, and we've partnered that with Gilead, and now it's actually in human trials as well. So that's kind of the tip of the iceberg. I think my point is we started this a number of years back, but I think it's even more important now, is it really is. Doing science and having the taxpayers pay for it, which I've been doing for 40 years, is a privilege, okay? Somebody pays me to do what I love. It's a privilege, and with that privilege, I think comes an obligation to give back to the people who give you that privilege. And I think one way we can give back is by using our discoveries to advance human health. And I think in doing so, we might actually begin to become independent of the challenges that we see right now in the federal funding of research, and that clearly are going to be there in the future. So that's the model we're building, and thanks for your attention.